Now let's have a look at two different approaches which are used in the immunofluorescence technique. The first one is the direct approach. In this approach, we use the monoclonal antibodies with the fluorescent indicators on them, and these antibodies are directly bonded to the protein or the cell or anything, the biological structure that we want to see under the microscope. Okay, but the problem is that these antibodies, monoclonal antibodies with the fluorescent indicators, are too expensive. They can be commercially available, but we have to purify our sample after treating the proteins with these antibodies, and it's energy consuming, time consuming, and it costs a lot. Within another approach, we can use the indirect immunofluorescence, which we use two uh, animal source antibodies, such as the mouse and the murine. And the idea is that we first treat our proteins with the primary antibody, which does not have the fluorescent indicators on them, but this antibody has the affinity towards this protein. In the second step, another antibody as the secondary antibody with the fluorescent indicator on that, which has the affinity towards the primary antibody, is introduced into the sample. Okay, and because of this linkage, we can appreciate the recognition of the proteins that we had in our mind. The advantage of this technique is that because these are the animal source antibodies, they are not too expensive on one hand, and on the other hand, several different of the secondary antibodies can be bonded onto the heavy side of the primary antibody, so the intensity of the light can be increased. In this case, only one antibody can be observed on the protein due to these antigens, but in this case several antibodies can be observed, and the light intensity that we get will increase. And again, I have to mention that this method, although it may seem that because two steps are needed, it will be more time consuming. Okay, it may be, but it is more cost effective.